of microbes, natural substrates of microbes are converted into value-added products. The microorganisms exploited are natural, laboratory selected, mutant, or genetically engineered strains. In general, microorganisms are capable of producing uh, producing an array of valuable compounds that ranging from macromolecules such as proteins and nucleic acids and carbohydrates to a smaller molecules called metabolites in small amounts. These metabolites are produced by such microorganisms are further classified as secondary metabolites and primary metabolites and secondary metabolites. The primary metabolites are produced by the microbes during their energy metabolism and are essential for the vegetative growth and development and reproduction of the organism. That includes the carbohydrates, vitamins, proteins, and amino acids. When coming to the secondary metabolites are produced after the growth of the microbes, after the growth of the microorganisms, and these secondary metabolites are usually produced once the microorganism is reaches to the a complete growth stage. Then, which includes antibiotics, alkaloids, and toxins. See here, as just we discussed, a primary metabolites that are produced by microbes during their energy metabolism and are essential for vegetative growth, development, and reproduction of the organisms. That includes the primary metabolite production process includes the protein production and vitamin, carbohydrate, and amino acid. When coming to the secondary metabolites that are produced after the growth of the microbes and usually during the stationary phase of the growth of the microorganism. That includes antibiotics, alkaloids, and toxins. The microbial population produces such compounds for their own benefits, but those are exploited scientifically to produce the products of potential applications in human therapies, and also used for many other uses. This could be made possible through genetic engineering techniques, as we already discussed about the insulin production is the best example, employing genetic engineering techniques by insertion of the human insulin gene into bacterial cell. The microorganisms also have the ability to produce the compounds that are not even produced in the laboratory. The microbial products such as enzymes, vitamins, alcohols, and amino acids, and recombinant products like recombinant proteins, fertilizers, and biopesticides, and economically important valuable products with a good market value. These products are cheaply produced by the application of microorganisms. For industrial production, the microorganisms are selected for their metabolic activities to produce one or more specific products in high rate. The microbes are also utilized in areas such as the unit which we are going to use, we are going to discuss in the next topic, that is water treatment and microbial enhanced oil recovery and biodegradation and biomining. With these aspects, microbial biotechnology plays an exemplary role in our lives and lifespans. So, what are industrial microorganisms? Here, microbes are used to synthesize a wide range of products that are highly valuable to human beings, such as the microbes are industrially exploited to produce the products such as food additives and beverages and therapeutics for human and animal health. The industrially exploited microorganisms are called industrial microorganisms. It is mostly included the bacteria and the fungi. There are endless microorganisms that are used in daily life at today's industrial level. For example, the Streptomyces gracious and Streptomyces parvulus. These two are the bacteria. Those microbial product is the streptomycin and actinomycetin D. The product is antibiotics. When coming to the amino acids is produced by the organism which employed industrially is Brevibacterium flavum and Corynebacterium glutamicum. These two produce the microbial product is lysine and glutamic acid. And the beverages like wine and beer is produced by employing the microorganism is Saccharomyces cerevisiae and Saccharomyces iravam. And the other one, organic acids, the microorganisms like lactobacillus species and gluconobacter suboxidants. These two are the microorganisms which can be employed in industries for the production of organic acids. The byproduct is lactic acid and gluconic acid. The other one, vitamins. Vitamins, the organisms like Trispora and Ashbay, Gossypii is produce lysine and glutamic acid. And when coming to the organic solvents like ethanol production and acetone production, both is produced by employing the microorganisms like Saccharomyces cerevisiae and Clostridium acetobutylicum. Clostridium acetobutylicum is the bacteria that can convert that can produce the byproduct is ethanol and acetone. Food products like cheese and yogurt, both two is produced by employing the microorganisms like Penicillium roqueforti and Lactobacillus bulgarius. Penicillium roqueforti, and Lactobacillus burgerius. These two microorganisms can be employed for the production of cheese and yogurt. When coming to the enzymes like amylase and cellulase is produced by employing the microorganisms like Aspergillus oryzae and Aspergillus niger. And cyclosporin, it is a pharmaceutical drug that can be produced by employing the microorganism, that is Trichoderma polysporum. Mostly here we discussed about the microorganisms like fungi and bacteria are widely utilized in the production, in the industrial production of the antibiotics, amino acids, beverages, organic acids, vitamins, organic solvents, food products, enzymes, and pharmaceutical drugs. When coming to the fermentation, here the fermentation is defined as a metabolic process in which an organism converts a sugar into alcohol. It is an anaerobic process. Here we will not use any oxygen, which means it requires the absence of oxygen. Industrial fermentation refers to the chemical process involving microorganisms to obtain several products of human and animal feed. It usually begins with a suitable microorganisms and maintenance of suitable conditions such as pH, oxygen control, foam control, and temperature. Usually, glucose is converted into pyruvic acid through glycolysis process. In the presence of oxygen, the pyruvic acid enters the TCA cycle, gets oxidized to produce 36 to 38 ATP molecules, but the absence of the oxygen through the alcoholic fermentation process, the pyruvic acid gets converted into ethanol and carbon dioxide. In lactic acid fermentation, it gets converted into lactate. These are the two main types of industrial fermentation techniques, such as solid state fermentation and submerged fermentation. What is solid state fermentation? The type of fermentation is carried out on a solid substrate such as sawdust, wheat bran, and cereal grain, cereal grain. The solid substrates are the source of nutrients that are required for the growth of the microbes. This type of fermentation does not involve the usage of liquid medium, means no water is required for the growth of the microorganism. But some moisture content is essential for this process. A single pure cultures or mixed cultures may be used this type of fermentation. The pre-treatment of the raw material is carried out to increase the bioavailability of the nutrients for the microbes to act on them. The pre-treatment techniques involved like soaking, boiling in water, and other chemical treatments. It is mostly used for the production of fermentation foods such as bread, yogurt, and cheese. The foods produced through the process are nutritious and easily digestible. What are the examples of solid state fermentation? Here, the production of cheese by Penicillium roquefort and production of alpha amylase by Aspergillus niger and the production of edible mushroom such as agaricus and pleurotus. The solid substrate commonly used for this process is sawdust. So when coming to the submerged fermentation, here the submerged fermentation, this type of fermentation technique that involves the growth of the microorganisms as a suspension on liquid medium called nutrient broth. It is also known as liquid fermentation and it utilizes substrates like molasses, soluble sugars, vegetable juices, sewage water and broths. It usually requires a high content of oxygen for the growth of the microorganisms. And it is mostly suited for bacterial cells and that require high amount of the moisture content. The microorganisms utilize the nutrients on the broth and produces bioactive compounds. The compounds that are usually secreted in the fermentation broth are harvested by different techniques which is subjected to centrifugation, is finally dried and packed. This submerged fermentation is mostly exploited for industrial production of enzymes. For example, production of citric acid by Aspergillus niger and production of lactic acid by lactic acid bacteria. One more time. Here we have the fermentation techniques are of two types. One is solid state fermentation and other one is submerged fermentation. In the solid phase fermentation, we discussed it happens in absence of oxygen. But submerged fermentation, it requires moisture. So what are the fermentation modes? Here we have the fermentation modes like a batch fermentation and a fed batch fermentation and continuous fermentation. So first coming to the batch fermentation. What is batch fermentation? A batch fermentation, it is a closed culture system. The microorganisms are usually inoculated into a fixed volume of medium that supplies the essential nutrients for the growth of the microorganisms. As the growth phase of the microbe proceeds, like there occurs the depletion of the nutrients, which is a disadvantage of the method. But during the stationary phase, the microbes produce metabolites that are extracted from the fermenter through downstream processes. The accumulation of metabolites also ceases the growth of the organisms in batch fermentations. When coming to the fed batch fermentation, it is a modified method of batch fermentation. The fermentation is the most widely used one these days. In this fermentation, the substrate is added in increments several times during the fermentation process to increase the concentration of the biomass. Since The substrates are added at different periods, like different intervals. A nutrient depletion is avoided during this process, and hence the growth of the biomass is maintained at optimal levels. The duration of the log phase and stationary phase of the growth of the microorganism is also high in this fermentation process, means a fed batch fermentation process. And hence the production rate of the microbial products or secondary metabolites is also high. These are highly utilized in the industrial processes. When coming to the continuous fermentation, in this type of fermentation, the fresh medium is added into the fermenter continuously or in between during the course of the fermentation process. Likewise, the used medium with the microorganisms are subsequently removed from the fermenter tank for the recovery of the microbial product. Sorry. Microbial product. So, the high productivity is the major advantage of this technique. And the volume of the medium and concentration of the nutrients are maintained at optimal level during the course of the fermentation process. These are the fermentation modes. Like here we have the fermentation modes like batch fermentation, fed batch fermentation, and continuous fermentation. When coming to the as we already discussed about the scope and applications of the biotechnology, like in human therapeutics, in agriculture. In agriculture, already in the last week we discussed about rhizobium, azospirillum, and azotobacter, and the fungal biofertilizers like mycorrhiza, and algal biofertilizers like azolla, and when coming to the food and technology, in last week we already discussed about the microorganisms which plays their major role for the production of fermented foods like Saccharomyces cerevisiae for bread production, bread fermentation, and the Penicillium roquefort for cheese, and Lactobacillus bulgarius for yogurt production, and Aspergillus soy for soya sauce, and Saccharomyces cerevisiae for cake. And we also we missed one point, that cake, a single cell protein. We already discussed about a single cell protein in the applications of biotechnology. Here the single cell protein that refers the total protein extracted from the microbial cell culture. That's what we are studying in this topic, microbial biotechnology, single cell protein in the microbial biotechnology. It serves as a nutritional supplement for both humans and animals. It is rich in protein content, but it also contains the carbohydrates, vitamins, and minerals. Hence, it is considered as a nutritionally rich supplement The several microorganisms like bacteria, yeast, fungi, and algae are utilized for the production of single cell protein. The substrates like molasses, whey, cellulose, animal manures are used for the production of single cell proteins. The microorganisms that utilizes these substrates as nutrients required for their growth and produce increased biomass concentration. Microorganisms utilized for single cell protein production are the bacteria, Pseudomonas fluorescens, algae, spirulina, fungi, aspergillus, and Aspergillus fumigatus, Aspergillus niger and Aspergillus fumigatus, yeast, Saccharomyces cerevisiae, and Candida utilis. Then what are the main applications of single cell protein is it is used as the supplemented food for undernourished children. It prevents the accumulation of cholesterol in the body. It lowers the blood sugar levels in diabetic patients. In environment, uh, when coming to the environmental biotechnology, in environment, the microbial technological principles are employed in a variety of processes such as bioremediation, microbial enhanced oil recovery, and wastewater treatment, biosensors, and bio-leaching. Here I want to give a brief discussion about the bioremediation. Bioremediation, it is the process of the removal of pollutants from the environment through the application of microorganisms. The microorganisms which utilized for the process are generally thermophilic, anaerobic microorganisms and are used to remove the organic waste from the environment. The microorganisms that degrade the environmental contaminants into less toxic forms through reactions that takes place as a part of their metabolic processes. Other increasing fact is that the microorganisms also possess some enzymes that allow some enzymes uh, that allow the to for the removal of the organic waste from the environment. The other interesting fact is that the microorganisms also possess the enzymes, organisms to utilize the contaminants as food for their growth. The effectiveness, for example, the Pseudomonas putida is the microorganism, the bacteria, which can able to utilize the oil waste, oil from the seawater that can utilize that oil product for their growth. In turn, that can be utilized for the bioremediation process. The effectiveness of the process that depends upon the environmental condition that allows the microbial growth and activity. The other term we widely used is biomining. Biomining, it is the process of cleaning up the series of the polluted with the series of sites that are polluted with the metals. It can also be referred as the process of extracting economically valuable metals from rocks, ores, and mine water, mine waste. There are two types of methods that can be employed in biomining. Those are bioleaching and bio-oxidation. Bioleaching refers to the removal of metals from low-grade ores and mineral concentrates. Usually, the metals are found that are binded with the surface of the solid mineral, and the microbes are able to oxidize that metal and make it dissolve in water. As the metals are dissolved in water, this method is called bioleaching. And this bioleaching method, bioleaching process, is usually done with application of microorganisms that are single-celled and undergo chemosynthetic metabolism, for example, mesophils. The microorganisms that grows in high pressure and temperature, a copper, zinc, cobalt, and uranium are the metals that are extracted by this process. On the other hand, some metals are not dissolved by the microorganisms. In such case, the minerals that are surrounded with the metals are decomposed by the microbes, allows the recovery of metals that are directly from the site. This process also involves the oxidation, but differences is that the metal of interest is solubilized. When coming to this is about the biomining and bioleaching processes and bio-oxidation processes. Here we discussed about biomining. Biomining happened with two methods that employed in biomining, such as bioleaching and bio-oxidation. When coming to the wastewater treatment is a technology, is also an application in which toxic materials such as textiles waste, textile dyes, heavy metals, and pesticides are removed by the microorganisms, and the diagnostics are developed to detect the disease-causing microorganisms in the water, so as to ensure the water quality. Here we employ the techniques like wastewater treatment to make the water into potable. And biosensors. Biosensors are the genetically engineered microorganisms are exploited as living sensors to detect the toxic chemicals in the soil and biological specimens like tolling a toxinogenic chemical found in chemical and radiation waste sites are also degraded by the application of the microbial population is called as biosensing or the organisms which used for this methodology or the methods are called biosensors. So what are the things now we discussed is a microbial biotechnology. It is a branch, it is a new emerging field of the biotechnology which involves the use of microorganisms for the production of the therapeutic and industrial products of economic importance. With its application, it is now becoming an in our inevitable branch of biotechnology. Several microorganisms are exploited for this process that may be a natural, a laboratory, and selected, and also genetically engineered ones. Usually, microbes are produced these products as a part of their metabolic processes, and these are now exploited industrially for large scale production. So the fermentation is the basis for industrial biotechnology. It is the process of conversion of sugars into alcohols. Fermentations are usually carried out in fermenter. There are two types of fermentation, such as solid state fermentation and submerged fermentation. As the name that implies, solid medium is used in the former and liquid medium used in the submerged fermentation. So the solid medium used in the solid state fermentation, whereas the liquid medium is used in the submerged fermentation. And batch fed fermentation, there are the different types of fermentation modes we used. Those are batch and fed batch and continuous fermentations. In human therapeutics, it plays an important role in production of insulin, human growth hormone, and tissue uh, plasminogen activator. These heterologous proteins are widely used in the treatment of diseases such as diabetes, dwarfism, and acute myocardial infections. In coming to the agriculture, in agriculture production of biofertilizers, pesticides, insecticides, and herbicides are its applications. These formulations are produced from microorganisms and are safe to humans and environment. These are cost effective too in food industry, a production of fermented food such as bread, yogurt, and cheese. In environmental biotechnology, biomining and bioremediation are its applications. Next, our topic is here. I'll try to share my. Here, biological wastewater treatment. Drinking water is also known as potable water. This potable water is initially held in a holding tank. Before going to tell about the water treatment, here we have water is the liquid source which can able to plays a major role for the living organisms, for the living um, things like humans and animals for maintain their daily life. So we can't expect our lives without the water. So as just we discussed about the microbial biotechnology, here we have the um, different sources which can able to pollute the water. So we have the biological techniques to control the activity of the pathogenic bacteria inside the water, and also we have the other techniques to kill the pathogenic bacteria which is presented in the water by different treatment methods called water treatment. Here we have two types of treatment methods. One is normal water treatment and sewage water treatment. Whichever it may be the treatment method, but here we are disinfecting the water and making the water into potable. What is potable water? Here, potable water is the water which is having all the nutrients that are used for the growth of the human beings and used for the metabolic process of the human beings, animals, and the living materials like plants also. So here the water treatment which has the drinking water is initially holded in a bigger tank called holding tank or a reservoir for settling the suspended matters. And then the colloidal materials such as clay are removed from the water by using the flocculent chemicals like alum. Alum means aluminium phosphate sulfate. After the flocculation treatment, water is treated by filtration, means a continuous agitation. A continuously agitation is uh, presented, that is passing through it through the beds of the sand or diatomaceous earth. And then some microorganisms like protozoans are removing during this flocculation method like filtration through the diatomaceous earth. Finally, this water is disinfected with the help of by passing through the chlorine. This chlorine kill the remaining pathogenic bacteria which is presented in the water. So, what is sewage? As just I discussed with you people, sewage and wastewater treatment. The wastewater is water after being used from the household or industries or anywhere else like hotels, everything, lab, is called sewage. Sewage includes like household, toilet waste, industrial waste, and rainwater. This sewage has to be treated before it is discharged into rivers and sea. Sewage treatment is done by the three steps. One is primary treatment, secondary treatment, and tertiary treatment. What are they? In case of primary treatment is the process of removing of solid matter called sludge from the sewage and about 40 to 60% of the suspended solids that can be removed by passing through the sewage through the sedimentation tanks. Sometimes flocculating chemicals are added to remove more solids. The primary treatment removes approximately 25% to 35% of the biologically oxygen demand of the sewage. What is BOD here? Here, the BOD is the measurement or the measure of a biologically degradable organic matter in water. A BOD is determined by the amount of oxygen that are required by the bacteria to metabolize the organic matter. So here we can use a BOD is the best measurement of biologically degradable organic matter in the water. When coming to the secondary treatment is the biological degradation or organic matter in sewage after a primary treatment. In this process, the sewage is already aerated vigorously with the help of the growing, with the help of aerobic bacteria and other microorganisms that can able to oxidize the dissolved organic matter to CO2 and water. Two commonly used methods of secondary treatment are activated sludge systems and trickling filters. What is activated sludge system? This activated sludge system that consisting of aeration tank where air or pure oxygen is mixed with the effluent from the primary treatment and some of the sludge from a previous batch is added to the incoming water. The sludge inoculum that which contains various number and a large number of metabolizing bacteria together with yeast, molds, and protozoa. Species of Juglio bacteria are important as they form flocculent masses in the tank and oxidizes the organic material. So most of the settled sludge of are removed from the treatment in an anaerobic sludge digester. Then the effluent is sent on for final treatment. The activated sludge systems remove a 75% to 95% of BOD from sewage. What are trickling filters? Here the trickling filters are the method where sewage is sprayed over a bed of rocks or modeled plastic. In the rocks that provide enough surface area for microbial activity and the space between the rocks and large enough to produce means provide air to the bottom. Since that sludge containing more amount of the microorganisms, so that we have to provide enough amount of the air to the bottom. A slimy gelatinous film of aerobic microorganisms that grow on rock surface and metabolize the organic matter in water. When it passes through into CO2 and water. Then we can say like trickling filters that can able to remove 80 to 80% of biologically like a BOD, biological oxygen demand. Here, see the trickling filter image as just we discussed what we have discussed here regarding trickling filters. In this method, sewage is spread over a bed of rocks and molded plastic. The rocks provide enough surface area, the space between the rocks and large enough to provide. Here you can see when the water is passed through the pump, the collection of the groundwater taken place through the rock. So the sewage drips on the rock bed. The rock bed with a slimy bacteria growth. Here you can see. And here the collection of the groundwater taken place. This is a rotating arm, and here the oxygen production takes place that can able to remove the 85% to 80 to 85% of the bacteria. Here a wastewater, when it passes through the tank, that removes the solids. When here the pre-treatment taken place, where the sludge is separated, then the next zone is aeration zone, then next one is settler, and here in this tank we have the sand for filtration, and the next one is disinfectant with the help of chlorine. Here you can see we use the membrane, that is the diatomaceous earth diatoms as a membrane to filter. So the final one is effluent is thrown from the tank. The pure water like potable water is treated with the help of disinfection. Then yes, we will get the 100% pure water. So the final treatment is usually the effluent from the secondary treatment is disinfected and discharged onto the land or into the water. Then it is subjected to the tertiary treatment. Tertiary treatment uses physical filtration and chemical precipitation to remove all the BOD material, nitrogen and phosphorus from the water. Tertiary treatment provides a drinkable water like potable water, whereas secondary treatment provides water for irrigation. And the final treatment, the sludge is further digested in aerobic sludge digesters. Bacteria degrade organic matter and produce methane and carbon dioxide gases. In the first stage of anaerobic sludge digestion, there is the production of CO2 and organic acids by anaerobic fermentation. In the next stage, organic acids are metabolized to form hydrogen and CO2 and some organic acids are metabolized to form hydrogen and CO2, and some organic acids like acetic acid. In the third stage, they are converted into methane by methane producing bacteria. In the wait, yes. Mm. If you can see the general overview of the um, plant, like a biological wastewater treatment, here you can see usually a raw wastewater influent. This is primary, and the primary sludge is primary residual is moves to the landfill, and in the primary clarifier, that is primary sludge, is moves to wastewater treatment residual, that is a bio solid process and disposable. In case of secondary treatment, that is biological treatment system, the primary sludge, attached growth and suspended growth and constructed wetland, and the secondary sludge is act like a wastewater treatment residual. The final step is disinfection step. Clean wastewater effluent discharge to the receivers, and the biological wastewater treatment, that to remove the suspended solids and dissolve organic load from the wastewater by using microbial populations. The microorganisms are responsible for degradation of organic matter that can be classified into three. That is aerobic, which requires the oxygen; anaerobic, that grow in absence of oxygen; and facultative, can proliferate their either in absence or the presence of oxygen. Just now, as just we discussed, if the microorganisms are suspended in the wastewater during the biological operation, the suspended growth processes or recycling of settled biomass is required. While the microorganisms that are attached to a surface over which they grow, that attached the growth processes, the biomass that attached to the media, that is rock, plastic and wood, the recycling of settled biomass is not required. Here, attached growth process, what is this process do means remove the nutrients and remove the dissolved organic salts and remove the suspended organic solids and remove the suspended solids. If you can see that a biological film, you can see the presence of the oxygen and wastewater. Those contains a biomass which contains the presence of viscous, a jelly-like substance containing bacteria on the filter medium. We used trickling filters and the rotating biological contract for this wastewater treatment. What does the trickling filters contain? When you see the trickling filters in a side view, you can see the presence of a rotating arm, a rotating distributed arm that sprays wastewater over the filter medium. Here you can see the rotating distributed arms, one either sides, left and right. The medium like rocks, plastic or other material. The water is collected at the bottom of the filter. Here you can see under drain. And then this is the actual structure of the trickling filters. Here you can see this is the side view. In the side view, rotating distributor arms are there, packing media. Actual the structure here, the filter media and the retaining wall. Here the outlet wall, where the outlet pipe is connected, where whichever the from here the influent is goes, the wastewater is goes, where the with this help of these arms, here one arm and this is another arm. This is called as distributor arms that always used to rotate with the help of the microorganisms, where we can filter. And here you can see the presence of ventilation port. From here the oxygen supply takes place. This is distributor base where the rocks are presented. See here, these are the trickling filters. You can see the waste, the collection of waste taking place here. Here the pure water, the wastewater is treating with the help of the distribution arms here. And the design and consideration of the trickling filter. The influent wastewater is wastewater characteristics and the degree of treatment anticipated for the removal of BOD and TSS, and the temperature range of applied wastewater, pre-treatment process, and the type of filter media, recirculation rate, hydraulic and organic loads applied to the filter under drainage and ventilation systems. We already have seen this trickling filter side view, no need of going again. And what are the filter media here we use is the crushed rocks, I have shown you already, and plastic media or random packing media and modular packing media. And crushed rocks are durable and those are insoluble. That can be available locally, and but reducing the avoid spaces for passage of air, the less surface area per volume for biological growth. Here you can see the filter media, that is cross flow and tubular and Paul rings. The systematic diagrams of modular and random packed media is used for fixing film treatments, that is reported by the scientists both Dax and Young. Why we need this filter media? Since the ideal filter packaging material that has a high surface area per unit volume, is low in cost, has a high durability, has a high enough porosity. So the clogging is minimized and provides good air circulation. A flow diagram for the trickling filters. Here you can see a recirculation, a portion of the trickling filter effluent recycled through the filter. A recirculation ratio R is equals to a returned flow by influent flow. Here you can see that this is Q, influent flow, the primary clarifier that removes the sediment and upper sediments, and the returned flow. Here returned flow is for recycling. This is the trickling filter that is moved around, and here the waste sludge collected, the final effluent is treated with a final treatment. Why we need recirculation? Means maintain the constant wetting rate and dilute the toxic waste and increase the air flow, and recirculation flow dilutes the strength of raw waste materials like raw waste water, and allows untreated wastewater to be passes through the filter more than once. A common range for the recirculation ratio is 0.5 to 3. When coming to the single stage, this is not important for you now. Here you can see the uh, um, a big tanks uh, uh, with trickling filters. When coming to the under drainage system, there are two purposes: to carry the filtered wastewater and the biomass lump that is called as a sludge solids from the filter to final clarification process, to provide for ventilation of the filter to maintain aerobic conditions. The underdrained system is generally designed to flow 1/3 to 1/2 full to permit ventilation of the system. When the trickling filter system, the air is supplied by the natural draft or forced draft fan that is rotated continuously. The forced draft fans have been applied in order to provide the adequate oxygen for the removal of the sludge. A stone media trickling filter design. Here we discussed about the biological oxygen demand that can be expressed as a kg meter cube by density. So that can be calculated the organic loading rate here. For example, influent BOD is equals to 200 mg per liter. The influent flow is 1.8 ml per diameter of the filter is 16 meter, and the depth of the filter is 2 m. So we don't need all these things here right now. So another thing here I want to discuss with you is Yes. As just now we discussed about solid wastewater treatment, and other thing I want to show you regarding sorry, this is not open. I don't know why. So, what we discussed here today means the types of pollutants here is suspended solids and dissolved solids and organic and inorganic materials like phenol, oil, bacteria, inorganic materials like salt, calcium, magnesium, silt, and the conventional pollutants measures the oxygen, biological oxygen demand, and COD, chemical oxygen demand, and DO, dissolved oxygen. When coming to the solid state, when coming to the solid state content and nutrients like phosphorus, nitrogen, and acidity, that is pH, and the bacteria like fecal coliforms like E. coli, and the temperature. These are the conventional pollutants that can be measured. And just now I told you dissolved oxygen and biochemical oxygen demand, biological oxygen demand or biochemical oxygen demand, and chemical oxygen demand, and total organic carbon, and theoretical oxygen demand. And what is biological oxygen demand? Oxygen is removed from water when organic matter is consumed by the bacteria. The low oxygen conditions may kill fish and other organisms. When coming to the chemical oxygen demand, the quantity of oxygen used in biological and non-biological oxidation of materials in wastewater. What happened? Wastewater, like the determination of chemical oxygen demand is used in municipal industrial laboratories to measure the overall level of organic contamination in wastewater. The contamination levels is determined by measuring the equivalent amount of oxygen required to oxidize organic matter in the sample. For example, BOD by COD ratio, the greater the ratio, the more oxidizable biological treatable waste is there. So the ratios rarely exceed to 0.8 to 0.9. When coming to the total organic carbon, TOC, that measures the wastewater pollution characteristics based on the chemical formula. The test methods use heat and oxygen, UV radiation, and chemical oxidants to convert organic carbon to carbon dioxide, which can can be assessed in 5 to 10 minutes. When coming to the theoretical oxygen demand, the wastewater generally contains a mixture of carbon, hydrogen, oxygen, and nitrogen that can be calculated using the stoichiometric equations. Consider both carbonaceous and nitrogenous oxygen demand. Main difference from the COD. Here the methods of treatment, as just we discussed, the clarification, sediment flocculation are used for the suspended and colloidal pollutants. Evaporation, reverse osmosis are used for dissolved inorganic pollutants. Oxidation synthesis by microorganisms is carried out by biological treatment for dissolved organic pollutants. What it requires, the biological process. The cell derives energy from the oxidation of reduced food products like carbohydrate, protein, and fats that requires microbes with the ability to degrade the waste organics, contact time with the organics, and the favorable conditions for the growth. Here, the objectives of the biodegradation waste water treatment is to stabilize the organic matter that is soluble and non-settlable, and to reduce the amount of dissolved phosphorus, nitrogen in final effluent. Here the microorganisms are the single-celled organisms that can perform all the life processes that are independent. As we discussed, just in case of domestic wastewater treatment, the objective of biological treatment is to stabilize the organic content to remove nutrients such as nitrogen and phosphorus. Here the types are aerobic, anoxious, anaerobic, and combined aerobic and anoxic, and anaerobic process and the pond process. Here, this process removes the attached growth, suspended growth, and combined systems, and the other process remove aerobic, maturation, and facultative anaerobic. As just aerobic process, activated sludge, we already discussed that. This is used for removal of carbonaceous BOD and aerated lagoons. Attached growth is trickling filter. Just now we discussed about the trickling filter, which is used for the removal of carbonaceous BOD and nitrification, and rough filters, the trickling filters with the high hydraulic loading waste, and rotating biological contractors, and packed bed reactors. All these are used for the removal of carbonaceous BOD, and combined suspended and attached growth activated biofilters. Those are trickling filter solid contact process and biofilter AS process. Series trickling filter AS process. How to remove the, what are the things here we can see during the biological carbonaceous removal in case of our aerobic oxidation taken place. Here you can see the organic matter and the oxygen. Here the organic matter is treated with oxygen and nutrients with the bacteria. Here what happens? A removal of CO2, NH, and new bacterial cells and other end products are produced. When coming to the endogenous respiration, here you can see the production of energized cells. The other thing is, these are the forms of nitrogen found in the wastewater, like organic nitrogen, ammonia, nitrite, and nitrate. And biological nitrogen removal, how it happens through the biological material is the factors which can able to affecting the nitrification, like temperature, substrate concentration, and dissolved oxygen, pH, and toxic and inhibitory substances. Here you can see the activated sludge process. There are two phases to biological treatment, that is a mineralization of wastewater, waste organic producing CO2 and water and microbes. The separation of the microbes and water taking place. Here you can see the activated sludge process. This is inlet where the aeration basin taken place, and this is the clarifier. What is MLSS? Means mixed liquor suspended solids. The suspended solids in the totally mixed aeration basin is liquid. And operational parameters in the activated sludge process. Sludge process is the nature and F by M ratio and dissolved oxygen, RAS, a reacted configuration, pH, reaction kinetics, a reacted hydraulics and nutrition. The activated sludge process, the monitoring flows, organic concentration and loads, and the solid concentrations and settlability data, oxygen, dissolved oxygen in aeration basin, and dissolved oxygen uptake rate, that is mg per liter per hour, that is DOR. This is about biological wastewater treatment. With this, I'm signing off today's class. Thank you.